Hey guys, this is Dr. Anna, and I am here to continue with the evolution module. And this is the second segment of that module. Stopped at the previous segment at this slide, which is about Darwin and how he came up with his theory of evolution. We are right at there when I was talking about the finches of the Galapagos. So after this amazing journey on the Beagle around the world, he came up with all these facts. He saw the fantastic variety of species found on Earth. When you live in just one place, like in Virginia, you will never be able to figure out how many different species in the world. He also saw that every single species has a huge population, but at the same time, he saw that these populations are usually stable because too many species for the amount of food which is available. So therefore, they had to compete for getting the food, cause a stable number in the population. He also saw because of that not all of the offspring survives, but the ones who are the best fit for that environment. And he collected enough data for his revolutionary hypothesis, which is nothing but natural selection. I have a movie about it here.
The theory of evolution was developed by Charles Darwin and published in his book On the Origin of Species. He explained that natural selection is the process behind evolution. Charles Darwin developed his theories after his adventures upon the HMS Beagle. In his travels, he observed that creatures found on the islands he visited were similar to ones found on the mainland, but appeared to be slightly different. It wasn't until he returned home that he came to the conclusion that species are specially modified to their environments, and that's why they differ. He developed 4 conditions explaining why this happens. Darwin's theory of natural selection by descent with modification is testable and observable fact. Experiments have been conducted in the wild and in labs. Let's dive deeper into Darwin's 4 conditions. Condition number 1. Individuals within a population differ. There are features that differ within populations of the same animal. In our case, the feature that varies between our giraffes is neck length. Some giraffes were born with long necks, some were born with short necks. Condition number 2. The differences are, at least in part, passed from parents to offspring. Darwin's descent with modification is the idea that offspring are fairly similar to their parents, with some genetic differences. Condition number 3. Some individuals are more successful at surviving and reproducing than others. In the case of our giraffes, the long neck individual did not acquire its neck by stretching to grab the leaves. Instead, individuals within the population were born with the neck length that was longer than others. Because the longer neck allowed them to reach the food that was otherwise unattainable, it gave them an advantage. Condition number 4. The successful individuals succeed because of variant traits they have inherited and will pass on to their offspring. Giraffes with the longer neck advantage are in better health and able to pass this feature to their offspring. Because this trait is more successful than shorter necks, more individuals in the population have it. Over time, this process can result in populations that specialize for particular environments and may eventually result in emergence of new species. In other words, natural selection is an important process, though not the only process, by which evolution takes place within a population of organisms. Let's review. Individuals within a population differ. The differences are, at least in part, passed from parents to offspring. Some individuals are more successful at surviving and reproducing than others.
Excuse me.
The successful individuals succeed because of variant traits they have inherited and will pass on to their offspring. That's all for now. Thanks for watching.
So now let's go through the evidence for evolution. The first one is the branching organization of life. They're just talking about that all living things organized into branching hierarchy of groups nested within group. The branching structure based on both anatomical and molecular structure. But if you just think about your own family, probably some of you have had the family tree, that it's extremely similar to basically the evolution, how different species develop from other species based on natural selection. So you can do the same thing for your family tree, which really is extremely similar to this. So even at the higher level, which means inside one species, you have this kind of branching organization of life. So this here shows you the molecular record and the branching organization. So comparing DNA and protein structures, everyone uses the same genetic code. Number of amino acids different from human hemoglobins. I want to show you this movie.
Recently, we have sequenced and compared the entire genomes of both baker's yeast and a worm. The genes used by the yeast, a very primitive organism, are primarily those that deal with the core biochemical functions that all organisms must perform. It was expected and proved true that the worm contains most of these same genes. But the reverse is not true. Since the worm is a more modern creature than the yeast, the worm genes that deal with multicellularity are certainly absent from the yeast. But more than that, these extra genes are directly derived from the primitive genes that provide core cellular function. A larger study reveals at the genetic level, newer genes are copiously created from more primitive forms in order to accommodate the requirements of more modern and more sophisticated organisms. Some proteins are found in every living eukaryote on Earth. Fruit flies and bats, octopi and hippos, protists and humans. One such protein is cytochrome C. It is found in the mitochondria, the energy-generating parts of all eukaryote cells, and without it, the cells die. Experimenting with yeast, researchers have shown that human cytochrome C protein works for the yeast as well as its own. Indeed, they have substituted genes from fish, birds, horses, insects, and rats into the yeast as well, and all of these genes produce cytochrome C proteins that work for the yeast too. But the gene that codes for this protein has been buffeted with sequence mutations over the billions of years that it has existed. Essentially, all of these mutations are silent, null sequence sections inserted in a gene that do not change the protein that is built from the amino acids based on these genes. But these mutations do tell the genealogy of the owners. For example, humans and chimps have exactly the same amino acid sequence for this protein. It hasn't mutated in the 6 or 7 million years since our lines separated. And our common gene is at most 10 amino acids different from all other mammals, confirming our close relationship with other mammals. The differences increase as we step further back in time and further from our own branch on the tree of life. The yeast itself has 51 amino acid differences from that of humans and chimps, and is one of the most distantly related organisms from humans having separated from our branch over 1 billion years ago. Researchers have discovered another point about cytochrome C protein. It is somewhat easy to make other proteins that serve the same function. In fact, there are more possible proteins that can perform this function than there are atoms in the universe. So why do all organisms on Earth use the same version? The answer is simple, really. We inherited the one we use from our common ancestor. There are sections of an organism's genome, called transposons, that have no other function except for inserting copies of themselves elsewhere on the genome. And there are many very well-known sequences that do this. 2 such sequences are signs, or short interspersed transposable elements, and lines, or long interspersed transposable elements. There are about 850,000 lines and 1.5 million signs scattered throughout your genome, accounting for nearly 30% of the entire sequence. While they are useless to the genome and sometimes cause significant damage, they are useful to our investigations, since essentially the only way for them to go from one organism to another is through direct DNA duplication and inheritance. Your lines and signs are given to your children. The parts of your DNA that make up your genes are relatively small sections scattered among other useful parts of your genome, as well as your lines and signs. Like fingerprints, the patterns recognizable in these non-gene sections are unique to individuals. They are similar in relatives and less similar in distant relatives. This is the basis of DNA fingerprinting. And since these sequences are passed down from parent to child, finding the same sequence in the same place in two different organisms is direct evidence that the two organisms have a common ancestor. Biologists have used this idea to demonstrate exactly how different species are genetically related. Here are two examples. 3 different identical sign sequences are found in the same genome location of whales and hippos, which are closely related, while camels and pigs, which are not closely related to whales and hippos, do not have the same sequence. The most numerous sign element in your genome is called the ALU. In the human alpha-globin cluster, there are 7 ALU elements, and each one is shared with chimpanzees in the exact same 7 locations, guaranteeing a common ancestor. When ALU elements are deposited on top of other ALU elements, the markers become a chronology of development. Very recent human ALU sequences have been used to trace historic and prehistoric human migrations, since some individuals have new ALU insertions that other individuals lack.
As you just saw it in the movie, I'm showing you one more time this amazing picture here, where you can see the human arm, the cat legs, the whale flipper, and the bat's wing. Remember, these are homologous structures, the same bones under the skin with extremely different function. Remember, a human is grasping, the cat is walking on it, the whale is swimming, and the bat is flying with it. But look at the bones. This is the carp bones, and look at they are yellow so we all can see it. Look at it. How cool that is. Then if you look at the hand bones, see the hand bones? Here is the hand bones, and then the head bones here, and then these are the finger bones. Finger bones. Finger bones. Look at the finger bones. And the radius and the ulna right here. They are very, very similar. I think it's so much fun to just look at this picture and see that it could not happen without evolution. Why would you have the exact same bone structures for so many different functions? So just to compare it with the analogous structure, I want you to make sure that you understand the homologies, different functions, same structure. On the other hand, analogous structures are what looking similar. They have the same function, but completely different structure. If you look at the bird wing, it has the bone structure, but then any kind of bug has a wing which is flying with, but it doesn't have any bone structures. So that just means that they have very different origin. They are not related to each other. They have no evolutionary relationship. So the number 3 is the vestigial structures. Now the vestigial structures are anatomical parts or organs that have lost most of all their original function through evolutionary processes. Like a couple of examples, it includes the human appendix, the pelvic bones in whales and snakes, and the reduced digit or diclaw in the mammals such as dogs and pigs. And of course, or tailbones. These are very common that we hurt our tailbone, and it's crazy because you don't even understand why do you have a tailbone. But it is a vestigial structure, and these features are often homologous to fully functional structures in closely related species, and this is highlighting shared ancestry. I have an amazing movie here.
Look down at your body. Some of that stuff doesn't do anything, and I'm about to call it out. Get ready. Hey guys, Trace here chatting from DNews in DC. Your body is filled with genetic and biological garbage. Not just the junk DNA we've talked about in the past, but actual structures that your body took the time to put together and they don't do anything. A couple of new studies just completed by researchers at Boston University and the University of Liverpool focused on our feet. Take off your shoe and try and bend your foot in the middle. The average human foot should only bend in one place, at the ball. But 1 out of every 13, at least, can bend a second joint in their foot, just like chimps. Everyone's got that joint, but a lot of us, we can't bend it because our bodies added a tough ligament once we started walking upright. Those with softer ligaments, they can still bend their foot, like me. Not Anthony, though, and he's jealous. And that's not all. That's just one of the things that you can do at parties. Genetic stuff we once needed that is now irrelevant or non-functional is called vestigial, a vestige of our evolutionary past. Leftovers, like the second foot joint, are all over in our DNA. The appendix was useful at one point, for example, but now it's just kind of chilling. Some primates have appendices and use them to digest tough cellulose, but we really don't eat that anymore. So now we don't need it. Over the generations of humans, it's become slowly less pronounced, but it's still present, like an evolutionary intestinal time bomb. Another is the vestigial tail. It's just one of those weird things that our DNA gives embryos as a genetic leftover. But usually, they're given the command to die by the genes while you're still in the womb. But rarely, we make it through birth with this vestigial extension. If you're still in doubt about evolution, then combine the tail with a condition called lanugo, or a fine growth of hair that can develop on a fetus, and it sometimes is present at birth. So we have tails, and we're covered in fur at birth, and somehow we didn't evolve from animals? Get out of town. We have a whole other host of vestigial items, goosebumps, tonsils, wisdom teeth, and other things left over from when we had fur. We had more primitive immune systems, and we used cellulose-based diets. My favorite, though, our third eyelid. We have a vestigial third eyelid, just like reptiles, guys. It was designed to keep the eyeball moist and to protect it. And now we put that baby in a corner, genetically speaking.
So the next one is embryonic history. All vertebrate embryos start out very similar. We have gill slits, just like the fish or other embryos. We have long tail, which is inherited from ancient ancestors. And so the embryonic development somewhat repeats the evolutionary history. And I have a cool movie right here about this.
Watching a fetus develop reveals much about the ancestral history of that organism. A human embryo in its third week is a featureless disc of cells until a furrow appears and plows its way across the top layer of cells. This is the primitive streak. As the primitive streak then begins to shorten, it leaves a trail in the form of a tube of tissues. This is the notochord, a stiffening rod found in embryos of all chordates. In primitive vertebrates, it persists throughout life as the main axial support of the body. But in higher vertebrates, the spinal cord replaces it. But because our developmental heritage, the spinal cord cannot develop unless the notochord secretes the right chemical signals. The genome includes the information necessary for the embryo to develop properly. But the genome has a history. It has been passed down through a chain of ancestors, unbroken since the dawn of life. And it still goes through the motions of creating many of those ancestors. 300 million years ago, our ancestors were creatures who laid their eggs in water. These eggs were small and round and contained yolk sacs to sustain the embryo. This is the first stage of a human embryo. But we notice that our embryo goes through a stage where it takes the form of a flattened disc. We can thank our reptile ancestors for this stage. When reptiles started laying their eggs with new hard shells on land, the embryos had to be supplied with a larger yolk to feed them through a long period of incubation. In order to accommodate this larger yolk, the embryo itself became a flattened disc squeezed between the yolk and the hard shell. The human embryo no longer needs a yolk sac, but even after millions of years, each human embryo is rolled out to form a germinal disc, reptile fashion, before rolling up again. There are many more times in fetal development in which evolutionary history plays a role. Human embryos all have temporary pharyngeal pouches which echo the gill pouches of our ancient fish ancestors. These pouches eventually became the structures that evolved from the gill pouches of fish. Structures that include the eustachian tube, middle ear, tonsils, parathyroid, and thymus. And of course, it is not just physical structures themselves, but also the genes which control the development. Many of these genes controlling the development of human embryo are exactly the same genes that control the development of other creatures. In fact, some genes, like the Hox gene, which regulate the location and shape of limbs and parts, are able to control the development of creatures as different as flies and mammals.
After this extremely interesting movie, I'm gonna finish the second segment of the evolution module, and I will continue it in the third one. I hope I'm gonna see you there, guys. Bye.